All right, so let's get into chapters 9 and a little bit of 10. I kind of meshed them together to go over some really important topics and try to break things down. So remember that there are three types of muscle tissue. We have cardiac muscle, which is found in the heart. If we were to look at it, it has lighter striations than skeletal, but there are striations present. There also have these things called intervertebral discs. You can see them if it's horizontal, you see those striations going in a horizontal pattern. Intervertebral discs go in a vertical pattern, and they're randomly placed. The idea is it allows for the tissue to intermesh or connect with one another, and it does aid in the heartbeat. So we'll go over more of that as we go through through this class. It is involuntary, which means you don't technically have control over it. Second muscle, smooth muscle. Remember, looking under a microscope, it just looks pink. There are no striations present. It is found in hollow visceral organs, and it also is involuntary. And the last is skeletal muscle. That's the one you think about when you're flexing. That's what gives you motion. It is voluntary, which means you have control, and there are clear striations present, which you'll see in another slide. So let's go over, like I always do, I give you the description, then I'm going to give you some functions and then properties. So the muscular functions include movement. Remember how bones played a role in movement? Well, the other portion that plays a role in movement is skeletal muscle. It's like a push-pull, and they require both. Posture. These muscles hold you upright. It's real important because in a bent over position or with somebody with bad posture, it causes the organs kind of to shift, if you will, and not be up to full functionality. Constriction of vessels and organs. So we haven't talked about it yet, but when we get into specific systems, there you can clearly see that there is smooth muscle that surrounds it and aids in the movement or possibly the stopping of the movement by constricting that smooth muscle. Respiration. There are muscles found between the ribs, and that aids in the lungs either inflating or deflating. Produces body heat. When a muscle contracts, it produces heat. And then, of course, protection. I mean, obviously this picture is ridiculous, but you can clearly see that adds an extra layer of protection. Now, properties, things that make a muscle, if they're not all there, then it's technically not a muscle. The first one's contractility. That means the muscle can shorten. Excitability. That means there is a stimulus and response associated with that muscle. Extensibility. That means the muscle can lengthen. And then the last one's elasticity. That means it can go back to its original shape. And again, you received, or you should have within your Canvas module, a sheet that kind of gives some of these terms so you can review them. Again, it's not a study guide, but at least it outlines these terms. All right, so like everything else in your body, muscles need some form of protection, some form of great protection. And that is these protective membrane layers. And there's three of them. One's not enough. So one covers the muscle as a whole. And think of it as like going to Walmart and getting like a roast. So on the whole roast or on the whole muscle surrounding it is epimysium. There's little circles you can see in looking at these images is a fascicle. Fascicle is a grouping of muscle fibers. So surrounding each fascicle is another membrane. It's called perimysium. And then lastly, every individual muscle fiber has a protective membrane, which is called endomysium. The epimysium becomes tendon and it attaches to the bone. It's called aponeurosis. And the movement of muscle or the contraction of muscle is stimulated by motor neurons. Remember talking about that. There is the image that I was talking about. Look at those striations. This is skeletal muscle. It's very clear and concise. You can see darker and lighter striations, and I don't mean those horizontal lines. That's where it's together, but I'm looking at the vertical lines. So there's the striations. The light striation is called an I band, and it's made up of actin. The dark striations are called A bands, and they're made up of myosin. Hold it, you know this is crazy. It's going to get even worse. So there's these terms that it's just repetitive that you'll be able to at least remember it and understand it. Let's look closer. There we see a sarcolemma. That is the plasma membrane surrounding muscle fibers. A myofibril is a bundle of protein filaments. Remember the proteins I mentioned with the dark bands and light bands, the actin and myosin, those are the protein filaments. So a myofibril is a bundle of protein filaments. And there is an increased number of mitochondria present because remember mitochondria produces ATP, and in things in your body that requires more energy like muscle contraction, there is going to be an increased number of mitochondria present. Let's look even closer. This is a myofibril. There's those dark A bands with myosin. There's the light I bands with actin. Now here's a few more terms. There is a Z disc. A Z disc is the halfway point on an I band. The H zone is a halfway point of an A band. And if you measure Z disc to Z disc, that is called a sarcomere. And to make up a myofibril, it's sarcomere attached to a sarcomere attached to a sarcomere. That is called a myofibril. The last term that you can see there is an M line. That is the middle of the H zone. It holds myosin in place. Let's look even closer. So you can see the myosin heads. That's the red colored image on the left side. You can see the actin binding sites. Those are those blueberries, if you will. That's the way I describe them. The indentation you can see, those are where the myosin head would bind to it. This is an example. I can tell you that the muscle is contracting just by looking at this slide. There's the image. You can see that the myosin head is crossing over, those are those vertical lines, into the actin. Okay, so that shows a cross bridge. It's going across, and we're going to get into it shortly. Two more terms, if all those weren't enough. The first one, sarcoplasmic reticulum, otherwise known as SR. Those are specialized smooth ER. They're found in skeletal muscles. They store large amounts of calcium, and it allows for the start of a contraction. T-tubules are something that looks like this. They offer more surface area for contraction. They also contain proteins, specifically triads of proteins, that will release calcium. All right, so let me set up the stage. So what it is look at this. So remember motor neuron. Now we're going to start with where the motor neuron, the very end of the terminal branch, and you see it's right next to the sarcolemma. That is where we're going to start, and then we'll get into the actual muscle for contraction. Okay, so keep that in your head. The first thing I need to explain to you, because it's a new term for you, is an action potential. And I want you to think of an action potential as an electrical spark. Without an electrical stimulation like that, without an action potential, things can't happen, including contraction. So think of it this way. So an action potential is going to come from the central nervous system to the peripheral nerves, eventually to a motor nerve, which will stimulate it. Okay, so keep that odd thing in half. Now I'm going to give you the steps, kind of go over them briefly. I'm uploading a drawing of me doing it, and that should help you significantly. All right. So first, an action potential is going to arrive at the terminal branch or the very end of the motor neuron. Because of that, the voltage gates open and calcium comes in, which changes the charge. And remember, whenever you change the charge, something big is going to happen, and it is called depolarization. Because of the calcium flooding in, acetylcholine, which is a neurotransmitter, is put into vesicles and travels to the very end of that motor neuron and is released by exocytosis. And because of those vesicles, it isn't changed. It's pure, if you would, acetylcholine. The acetylcholine is going to cross the synaptic cleft, that is the space between the neuron and the sarcolemma of the muscle, and it's going to bind to specific receptors on that sarcolemma. Because that acetylcholine binds to the receptor, it causes potassium to go out and at the same time sodium to go in. So the potassium is going out of the muscle and the sodium is coming into the muscle. Step 6 is what I affectionately call the stop step, because think about it, as long as acetylcholine binds to that receptor, the transmission continues. However, if you take an enzyme, and the enzyme is called acetylcholine ester or ACE, and it binds in place of the acetylcholine, it blocks those receptor sites. The next series of reactions for contraction is called EC coupling, excitation, contraction, coupling. So EC is proper term. It's going to happen mostly in the T-tubules. So what's going to happen is that action potential is going to continue down the sarcolemma, and then it's going to enter that T-tubule. All right, so upon entry of that T-tubule, it's going to come in contact with those triads of protein, and because this is like that electrical volt, it's going to cause the triads of protein to change shape and release calcium. Calcium is going to travel and it is going to bind to a protein called troponin. Because it binds to troponin, it causes tropomyosin to move and unblock binding sites on actin. So now that opening on the blueberry, if you would, is open and a cross bridge can occur. The last steps associated with contraction is the cross bridge cycle. So this is the first step of that, is where the myosin head is in the actin binding site. They're clearly open. Attached to the myosin head is ADP and phosphorus. The second step is called the power stroke, and the energy in the myosin head is going to be released. It's utilized, causing the myosin head to shift, which causes the actin to move or slide, and ADP and phosphorus are released. The power stroke is where true contraction occurs. The third step is where the myosin head is removed or detaches from the actin binding site. ADP and phosphorus recombine to form ATP. ATP binds to the myosin head and it falls, if you will, downward. The last step of the cycle is going to revert back to normal. So the ATP is hydrolyzed. You have ADP and phosphorus on the myosin head, and the myosin head is going back to attach to the actin binding site. This is going to continue as long as all the calcium necessary is available and there is energy in the form of ATP present. This is just a demonstration at the fact that you don't need just one nerve, one motor neuron to cause a contraction. You need several innervating the myofibrils for this to occur. This is an example of a muscle twitch, and everybody's had like a twitch before or repeated twitches in a row, like your eye, and they say it's stress related. So a muscle twitch is a response of a muscle fiber to a single action potential with a motor neuron. And this could be done in multiple. If you think about it, if it just happens one time, that's a single twitch. But what happens if the twitches or the action potentials are coming in rapid succession? There you have a situation, instead of a nice curve on the top, you have something like, what happens is that muscle contraction never hits true peak, and it never gets a chance to relax. So it's not as strong as it could be. On that top curve, you can see there's really three portions of that wave. You have a lag phase, which means it's at rest and ready for stimulation by an action potential. You have the contraction phase, and then you have the relaxation phase. That means it's going back to its normal levels, normal charge, and then it's ready for a new stimulation by an action potential. This is to demonstrate the size principle of recruitment, or the size principle of muscular contraction. Recruitment is how many muscle fibers are involved with the contraction. So the idea is size does matter and amount does matter, but the size of the myofibrils and the number of bigger sized myofibrils will constitute a larger or stronger contraction. So size does matter in muscular contraction. Two types of contraction. The first one is an isotonic contraction. An isotonic contraction changes length, and when it changes length, it develops force to lift a load, which is your weight. So an example of this would be a squat. You get the length of the muscle shortens, develops that force when you explode up to lift that load. The second type of contraction is an isometric contraction. There is no change in length, still it has tension. Okay? Think of a wall sit. Imagine if I left you in that position, that correct position, for 2 minutes. Your legs begin to shake. You're going to be sore. Okay, that's an isometric contraction. There are three ways that allow for the ATP for contraction to occur. And I'm going to go over them just briefly. First one is direct phosphorylation. It's the use of creatine phosphate with ADP to create creatine and ATP. It is catalyzed by an enzyme, creatine kinase. It lasts for a very short period of time, anywhere between 10 and 15 seconds. It only produces 1 ATP per creatine phosphate. It is anaerobic, therefore no oxygen is required. The second one is called glycolysis and lactic acid formation. Lactic acid is a byproduct of anaerobic glycolysis and anaerobic metabolism. It occurs during stressful exercise. Lactic acid is useful to fuel the heart, but an abundance of lactic acid in skeletal muscle slows contractions. It's not as fast as direct phosphorylation. However, it is another anaerobic format, so therefore no oxygen is also required. The third and final one is aerobic respiration. It requires free oxygen. It is set of metabolic reaction. It takes place in cells of organisms to convert chemical energy from O2 or nutrients into ATP and waste products. It takes the longest to occur. It says about an hour, but I think it's a little bit less. Again, that depends on you. And it's going to produce 32 ATP per glucose. So this is going to produce the most ATP, but it requires the most amount of time. So we're talking about the difference between short duration runs or long duration runs. And you can see in a short duration run, you need ATP quickly. In a long duration run, you would need the ATP quickly to get started. Then eventually aerobic respiration will take over. These are some factors that increase the force of skeletal muscle contraction. Large number of muscle fiber recruitment. Remember I said the more you can recruit, the more fibers involved in the contraction, the stronger it is. Again, remember the size principle. Larger muscle fibers, the stronger the contraction is. High frequency of stimulation, that means the amount of action potential stimulating the contraction. And then imagine the more stretch and elasticity to a muscle, the stronger the force of contraction. With muscle contraction velocity in skeletal muscles, it can be broadly separated into a twitch, which are short bursts of stimulation caused by contraction during skeletal muscle contraction. It's so brief, muscle begins relaxing before reaching peak force, which I showed you with that very interesting wave, which looks something like that. Tetanic contractions are sustained muscle contractions when the motor neuron that innervates skeletal muscle emits action potential at a very high rate. Exercise. This is just to explain that exercise is necessary in two different formats and really a combination of both to exercise all muscles. So aerobic respiration is going to exercise your heart. That's running, biking, anything that gets your heart rate up. Resistance training is lifting weights or working in the machines on the gym that has some kind of weight that you're going to do, you're going to try to move. A balance between the two means you need to not only do aerobic respiration, but you also have to incorporate resistance training as well. All right, smooth muscle contractions. Just real brief on this, there are no T-tubules and there's very little sarcoplasmic reticulum, and I know you're like, please, not another 14-step memorization. No, it's actually very short and much easier. So in smooth muscle, you have these things called caveolae. It means little caves. They're found in the plasma membrane and they act like a passageway and they allow for the domino effect in calcium movement. So here's how it goes. Oh, look, calcium gates. That looks very familiar. So what happens is calcium is going to enter the cytosol or the cell itself. Calcium, remember, has a +2 charge. We're going to change the charge. Something big is going to happen. Remember, the sarcoplasmic reticulum contains large amounts of calcium, and that's where the calcium is going to come from to enter the cell. Next, the calcium is going to bind and activate a protein called calmodulin. That activated the activated calmodulin is going to activate myosin. You know what myosin is. Light chain kinase. It's an enzyme, and enzymes facilitate a reaction. So far, calcium gates, which you've heard before, calcium gates are going to open, change the charge. Then you're going to have the calcium bind with calmodulin, turning it on. Then the activated calmodulin is going to turn on kinase enzymes. Looks like something very simple. This looks like what we're talking about. Now we're going to go through the process very simply of going through cross bridges. You don't have to write out all the steps because you've already known it. Told you it would be simple. And this is just an image so you can see the cross bridges. These two slides that you'll see is a compare contrast between the three types of skeletal muscle. Just review it. It'll help you in your understanding, but the comparison between the three. Here's that second slide illustrating the differences between the three. So muscle tone is continuous contractions that are not full force that you cannot see. All you see is muscles right there. You can say he's well defined. However, the reason you can see it is very small contractions that you even can't feel as the person with the muscle tone. All right, last little bit. So skeletal muscles are named by location, like, so this last one is how they're named. Location, where is it found? Temporal bone. Shape, deltoid, means kite. Size, gluteus maximus versus gluteus minimus. You can bet that gluteus maximus is larger. Direction of fibers, rectus, up and down. Number of origins, biceps, 2, triceps, 3. Location of attachments, what do both ends attach to? Sternocleidomastoid, sternum, mastoid bone. And then muscle action, abductor, adductor, move in and out of the limbs. And then muscle action, flexion means to get smaller. Extension means get longer. Abduction is going to mean middle, away from midline, a limb going away from midline. Adduction is the opposite, a limb going towards midline.